Let's talk about the enzyme-substrate complex. So first, we'll talk about activation energy. And any reaction, you need to understand, has an initial energy barrier, and we call that the activation energy. And the reason this energy barrier exists is because before new bonding can happen, any existing chemical bonds between the reactants need to be broken. So for instance, if, say, a reaction wants to use this circle atom and this square atom, we're using our imagination here, these two things have to be broken apart in order for them to be used in other reactions. And so the activation energy is the energy that it takes to get over this hill, hypothetically speaking, of energy necessary to break up the chemical bonds that exist between the reactants that are going to be used in a new reaction. And so we use big E, little a, to represent activation energy. And the rate of a reaction is determined by the activation energy. So that means that the higher the activation energy, the slower the reaction is going to proceed. Enzymes are proteins that act as what we call catalysts. And catalysts make a reaction happen faster by lowering the activation energy without actually being changed themselves. And so if we were to imagine another graph like the last one we saw, instead of an activation energy being high like this, with an enzyme it can be much lower, such as right here. And one example of an enzyme that causes reactions to be catalyzed is this here, which is what we call lactase, which is the enzyme that allows you to digest milk sugar, which is called lactose. If you do not have this enzyme, you're probably lactose intolerant. So the molecule that an enzyme interacts with is called a substrate. And so in this picture here, what I've circled is the substrate, and this here is the enzyme. Now an enzyme's active site, which is here, is the location where it interacts with the substrate. Now the enzyme and the substrate fit together in what we call the enzyme-substrate complex. And when the enzyme's finished doing whatever it is that it does, it releases products. And in this case, the substrate started out as being two things joined together, and the enzyme split them apart, as you can see here. Now some enzymes break substrates apart into smaller pieces, such as this one that we were talking about a minute ago. Other enzymes actually put molecules together to form one or more other molecules. And so you need to understand the parts of the enzyme-substrate complex, the location of the active site, and what happens when enzymes interact with their substrates.